All right, everybody, here we are with the electron transport chain, also known as aerobic metabolism. We've broken this down into two different components, the first of which we're going to talk about the transport of electrons and the proteins that are involved in the transport of those electrons. The second part of this discussion is going to be focused on actual synthesis of ATP. So we will go ahead and begin and go from there. So your introduction. Well, first and foremost, it's important to understand and appreciate that aerobic organisms are sustained by huge amounts of ATP, adenosine triphosphate, that they can generate. Now, this is due to their ability to use oxygen, O2, as a terminal electron acceptor. Now, there are several properties of oxygen that make it very favorable and unique for extracting energy from organic molecules. And these organic molecules, keep in mind, we're focused on, well, molecules derived from glucose. Basically, we have a chain where we started with glucose, went through glycolysis, went through the citric acid cycle, and generated molecules, NADH and FADH2, that we can extract energy from. Now, what makes oxygen unique is that it's found almost everywhere on Earth. It diffuses very easily across membranes, which is unique, and we'll talk about diffusion across membranes a little bit later on when we talk about membranes. But it's highly reactive and it can very readily accept electrons. Now, NADH and FADH2, those are our money makers, because these are molecules that are produced in glycolysis and the citric acid cycle, aka TCA cycle, that generate usable energy in the electron transport chain. So the electron transport is the process by which reduced coenzymes are oxidized. Enzymes that catalyze reactions have the ability to simultaneously create a proton gradient across a membrane. So this is pivotal right here, a proton gradient. So when you think about a gradient, I want you to think about high concentration and low concentration. And a gradient implies a gradual movement of those concentrations, but I want you to focus on the keys of high concentration versus low concentration. Now, there are a series of redox carriers that receive electrons from NADH and FADH2 molecules. So it's the generation of these two molecules that, well, they're acting as electron carriers. So whenever we produced NADH in glycolysis and FADH2 and NADH in the citric acid cycle, well, really all that we were worried about was their ability to hold on to electrons. Now, ultimately, electrons and protons are donated to oxygen to form water. Now, oxidative phosphorylation is the process through which electron transport is coupled to ATP production. So I want you to think about the two words here, oxidative phosphorylation. Oxidative implies that there's an oxidation and reduction taking place. And this phosphorylation, well, phosphorylation we've talked about in the form of transferase enzymes that transfer phosphates onto adenosine diphosphate. So you're phosphorylating something. So it's through this oxidation reduction that enables phosphorylation to take place. Now, what we really need to focus on for all of this is that little membrane-bound organelle, the mitochondria. This consists of an outer and an inner membrane. The outer membrane is fairly permeable. Think about the outer membrane, or the way that I always kind of think about the mitochondria is I think about if you ever buy oranges in those kind of mesh bags, the outer membrane would be analogous to those mesh bags. So they're going to have pores that are fairly large. But is everything going to be able to squeeze through there? No, because something simply won't fit into those pores. So this outer membrane, well, it's fairly permeable. The inner membrane would be much like the rind of the orange, or the Is it a rind? Yeah. Yeah, the rind of the orange. Virtually nothing's going to get through there. And that inner membrane is that orange rind. This is impenetrable to charged molecules. Transport proteins are needed for actually crossing this membrane. So there has to be something there to facilitate the migration of molecules across that membrane. Now this is going to contain enzymes needed for electron transport and oxidative phosphorylation. So when you think about the electron transport chain and oxidative phosphorylation, think about most everything is functioning, or our gradient is basically across that inner membrane. It's not so much the outer membrane. Now, within the mitochondria is the mitochondrial matrix, which contains all but one enzyme of the citric acid cycle. The enzyme that it does not contain is the succinate dehydrogenase complex. Here's an example of our mitochondria. We've got our citric acid cycle taking place on the interior of the mitochondrial membrane. We've got pyruvate, which can make its way across that outer membrane and then is transported into the inner membrane. NADH, FADH2, these are all going to be produced within or on the interior side of the mitochondrial membrane, inner mitochondrial membrane. Now, coupled processes required for ATP synthesis. So that means we have multiple processes that are kind of paired up with one another that enable ATP synthesis to take place. Now, respiratory electron transport chain carries electrons from reduced coenzymes to oxygen. So our reduced coenzyme is where electrons are being carried from, ultimately, to oxygen. The electron transport chain releases energy. The proton concentration gradient is used to provide energy for the phosphorylation of ADP to form ATP. So these two processes are paired up with one another to basically take place. Now, your overview of the electron transport chain is shown below. I'd like to draw your attention over here to this green blob, brown, yellow, and purple. What we have here are the different complexes, protein complexes. We've got complex 1, complex 2, complex 3, complex 4. Now, the transport of electrons occurs through 4 different complexes. Electrons are carried from 1 and 2 to complex 3 via this molecule known as coenzyme Q. Now, I'm going to get into the details of this in just a little while, but I'd like you to kind of focus on movement at this point. Now, electrons are carried from 3 to 4 via this molecule right here known as cytochrome C. So electrons are transported from the protein complexes using coenzyme Q and cytochrome C. So electrons are not directly transferred from one complex to the next. Instead, they kind of need an intermediary to do that. Now, the difference between these two intermediaries is coenzyme Q is, look at that, it's within the lipid bilayer, so it's within that membrane, whereas cytochrome C is on the inner or within the inner membrane space, inside or on the interior of the mitochondrial membrane. Now, electron transport, so the movement of electrons, releases energy. The electron transfers through the electron carriers is in the direction of increasing reduction potential in order of increasing affinity for electrons. Now, a recap on your reduction potentials. Your reduction potentials say or give you an idea of the ability of a molecule to accept or donate electrons. Electrons are preferably going to move from negative to positive. Okay. Now, the free energy of this can be summed up with delta G equals negative N, number of moles, times F, Faraday's constant, times delta E, the change in free energy. So the more positive that your delta E is, the better of an oxidizing agent. The more negative your delta E is, the better it is as a reducing agent. Now, if you look down here, we've got our transfers. So complex 1, electrons are transferred from NADH to coenzyme Q, or UQ, which is ubiquinone, which is another name for coenzyme Q. Electron transfer succinate to UQ is step 2. Step 3, electron transfer from FADH2 to cytochrome C, and then electron transport from cytochrome C to O2 are all displayed down here. And as you can see on this slide, all of your delta G values, the transfer from NADH to UQ, succinate to UQ, FADH2 to cytochrome C to O2, all of these delta G values are at very least negative, and some of them are hugely negative, so it's all favorable moves for those electrons.